Welcome everybody to today's edition of our webinar on emerging topics in biomolecular magnetic resonance. We have two truly exciting presentations again today, and both speakers agreed to recording, so we will record the talks and upload them to the ICMRBS YouTube channel. Oops, what did I click on? Something wrong. So here. Just to remind everybody, please use the Q&A box for putting in your questions, or raise your hand, and then we can unmute you that you can ask your questions yourself. There's also, please be reminded that we have the informal session afterwards where you can engage in discussions with the speakers or the panel. And there's the early career researcher edition of the ICMRBS webinar on magnetic resonance, so please tune in there.
Thank you. And it's a great honor and pleasure for me to introduce Thomas Prisner, the next speaker. Thomas is very well known in our magnetic resonance community for his seminal scientific contributions in the field of EPR spectroscopy and dynamic nuclear polarization, for which he received several prizes, among them, for instance, the Savoysky Prize or the Brooker Prize Lecture of the Royal Society of Chemistry, and many others. But we all know him also for his engagement in the magnetic resonance community. He's part of several boards, and currently he's the president of the Euromar Board of Trustees. And until 2020, Thomas was also president of the International EPR Society. So I would like to summarize briefly his background. Thomas studied physics in Heidelberg, where he learned magnetic resonance from very famous people like Karl Hauser and Peter Dinse. And then he graduated in Dortmund with Peter Dinse on pioneering work on Fourier transform and stochastic ESR. Then 1989, he went to MIT as a postdoctoral fellow with Bob Griffin, and where he set up the first 140 gigahertz EPR and DNP spectrometer there. He returned to Germany, to Berlin in 1919, and habilitated with Klaus Möbius by developing state-of-the-art high-field pulsed EPR. After his habilitation in 1996, he got immediately a full professorship in Frankfurt at the Department of Physical Chemistry. And now I would like to say I joined Thomas' lab a few years later in 2001 for habilitate, and since then the intellectual interaction with Thomas shaped also my scientific career, having several shared collaboration, also shared grant initiatives, and so this is a pleasure to be here, Thomas, for this lecture. So Thomas' research interests are very broad, from state-of-the-art developments in pulse EPR spectroscopy for studies of biomicromolecules in vitro and in cell, to dynamic nuclear polarization and hyperpolarization for NMR and MRI. The title of the talk is New Pulse Dipolar EPR Spectroscopy Experiments on RNA Molecules, and we are all looking forward to this latest developments. Thomas.
Many thanks, Marina, and also the organizers to invite me here. It's a pleasure. So let's open my presentation. Okay, I hope you can see my screen. Because this is a biological oriented audience, and because I was asked to keep the talk short on a 20 minutes time scale, so I decided to concentrate on one biological application. So what I will show you is not so much about pulse EPR methods development. I have seen there is a number of EPR people also attending here now, but more on what can you do with that on an RNA aptamer, which I'm looking for. So one of the methods which became very popular in recent years in EPR spectroscopy is the so-called Peldor-Dier methods. Peldor stands for pulsed electron electron double resonance, and here means the same, double electron resonance. And there are a number of methods which can measure the dipolar coupling between two electron spins. This is the most popular one, this 4-pulse version, which was developed from Hans Wolfgang Spies together with Gunnar Jerschke. And then it's a very simple sequence for NMR people to also understand. We just look on a refocused Hahn echo, which is the 1 electron spin which we are looking on. And then we recouple here because this is a weakly coupled dipolar coupled spin system. So the difference in Larmor frequencies is larger than the coupling strength. So we recouple all the interactions, and then we decouple, and then we recouple with the red pulse. We just invert the second spin within the experiment, and then we measure the dipolar interaction. And you do this as a function of moving this pulse. This inversion pulse for the second spin, which we call B usually. And then what you see is an oscillation, and the oscillation tells you immediately the dipolar frequency. Yeah, of course we are measuring in frozen powder samples. This is not a liquid state experiment. It's done in frozen solution, so we measure a powder peak pattern. And if by Fourier transforming this, you get the peak pattern, measuring the electron-electron dipolar interaction, and from that you can get the distance, of course. And usually what you do is, instead of doing a Fourier transform, you make a regularization of this integral formula, and then you get directly the distance distribution function for your biological system of interest. Now, we work a lot on RNA molecules with that methods. Usually a lot of measurements are done on proteins. That was what the method was developed for, or let's say where the first biological applications were looked on. Now, usually in biological systems, you take this MTSL spin label, which is quite flexible, but it can be very easily attached to cysteine, which you mutate on your protein, and then you make this pairwise, and then you measure the electron-electron dipolar coupling, and the range you can measure on is about 2 to 10 nanometers. Yeah, so 10 nanometers, you have to do some special tricks, but in principle, it can be done now, even for larger distances. Now, what you see here is, because this spin label is so flexible, you get, of course, a distribution of distances, because different rotamers of the spin label will actually, even if the protein is totally rigid, you will get a distribution of distances. So typically you get a resolution which is about 3 angstrom. So X-ray people would not say this is a very high resolution structure. And of course, it's not a structure at all, because you measure 1 distance. And if you want to measure a second distance, you have to make a second pair. Very different from the experiments we saw before. So here you then don't get 350 or 5,000 NOEs. You get 1 distance. Yeah, that's what you get usually with it. Now, we started to work many years ago. We started to work on that for RNAs. In RNAs, of course, for smaller constructs, you can synthesize them directly on a synthesizer, a solid phase synthesizer. And then you have to think about different labeling strategies. And we collaborate already very long time, very successful, with Snorri Sigurdsson from Iceland, an organic chemist. And he designed a lot of spin labels, but one of them, which we like very, very much, is this analog of a cytidine. Yeah, so it is an extension, which is the paramagnetic part to it. If you reduce it, it's actually a very nice fluorophore, so you can use it for fluorescence spectroscopy. In the paramagnetic state, the fluorescence is quenched, but then you can use it for EPR spectroscopy. And in a double-stranded helix, it's like a cantilever. It is absolutely rigidly incorporated. So that means there is no internal flexibility of the spin label. It fits in the major groove of the DNA and in the minor groove of the RNA, and there is no internal flexibility of the spin label. So all of the distribution of distances you see now, they are coming from the biomolecule itself. So this is, of course, an advantage. Now, there is also a price for that. First of all, the spin label is much more demanding to be synthesized. And it's actually demanding to incorporate it in large biological relevant RNAs. There are other strategies to do that. Now, there is another point. Because the spin label is so rigid, then, as we all know, the dipolar interaction depends not only on the distance, but also on the angle of the interconnecting vector with respect to the external magnetic field. And now, because the spin label is not flexible anymore, you don't average this angle anymore, or let's say you don't get a distribution of these angles anymore. And that means you don't expect to get a powder pattern. So instead, you will get this orientation information also encoded in your time traces. Now, what you do then in this case, you get additional information. This is great. It's not only one parameter, but you get also the Euler angles. But of course, then the analysis is more demanding. What you usually do, you simulate the Pelton time traces by assuming a geometry. So how can this be done? Just for the introduction, and then I'm over with that. If you go at the nitroxide spin label has two anisotropic interactions. The one is the nitrogen hyperfine interaction, which you can resolve at low magnetic fields, 0.3 Tesla, that's X-band. And the other one is the G anisotropy, all the three different G values, which you can resolve if you go to magnetic fields above 3 Tesla. And so at high field, you can now select different orientations with your pump and probe pulses by just choosing the different positions of your spectra. And at low field, at X-band, you can again select different positions with your pulses. This is just what kind of orientation of the spin label you excite. You know this exactly. We know all the hyperfine coupling parameters. And then we can make this experiment now at different probe frequencies. And this is shown here. And you see immediately how the dipolar oscillation goes from the single frequency to the double frequency. So if you move over here, you see really the 3 cosine squared theta minus 1 evolving, yeah, which gives you a -1 and a +2. And that means if you analyze this data, as shown here with the red traces, then you know everything about your biomolecule, because you know how the two spin labels sit with respect to each other. And because they are rigidly incorporated into the RNA or DNA molecule, so you get a lot of information on this. But it is, of course, a more demanding procedure. So we found over the years, we found a way how to do this. If you go to intermediate magnetic fields, this orientation information goes lost. This is a Q-band, and then you get a distance, and then we know already the distance, and then we go to low field, and then we get the out of plane angles, and then we go to high field, and then we get the rest. So all the 6 parameters can be disentangled. That this is true, I show you just an example. This is a simulation, of course. Let's assume the 2 spin labels sit in a way like this, or in a way like this, or in a way like this. So 3 different geometries, but with always the same distance of 3 nanometers. And then you see this pattern, which comes now from these different frequencies. They all look very different. So you can very easily, if you look on this pattern, you can immediately extract the orientation. What we did actually, we have a library of all possible orientations. And then what we do is we fit our experimental data to this library. Yeah. So this is a very, very nice way to get all the Euler angles, the polar angles, and the distance out of your data. Now, we used it over the years for many different applications on RNA and on DNA. And I just highlight here on this one slide a little bit what you can find. I'm not talking about that because we have done in-cell experiments with this kind of spin labels on RNA and DNA. And we found actually differences in the structure in the cell and outside of the cell, even for very simple motifs like double-stranded DNA or RNA. And if you're interested in that, we have already published part of it, but there will be also new data shown by Alberto Colauto, a former postdoc in my group, who is now in Imperial College in London. And next week there is a workshop on in-cell in Marseille, and I think you can still sign up if you're interested. And what we also did is we compared now our data with molecular dynamic simulation on these molecules. And again, we have already published, but we have already again new data that we can distinguish different force fields developed for DNA and RNA and show how good or how bad they correspond to what is the reality, which we believe is our experiment. This is a parameter-free fit and not a fit, a simulation, which we compare with our experimental data. And again, if you're interested, in 2 weeks or 3 weeks from now in the Euromar conference, Maximilian Gauker, he's a PhD student in my group, he will talk about that. So I'm not going to talk about that. We can of course use this to investigate the conformational dynamics of nucleic acid motifs in rather detail. Look about their dynamical motion. We don't see the dynamics because we freeze the sample, but usually for most of these molecules, we freeze the ensemble of all the possible states. And so there is also an advantage of freezing the sample because in this case, we don't have to worry about averaging timescales. We see all the possible conformers. We have investigated stacking of nucleic acid and their dependence on ligands or on ionic strengths. And actually with this rigid spin label, different from the flexible ones, you can actually do the experiment at room temperature if your biomolecule is big enough. Big enough means the rotation of your biomolecule has to be slower than the coupling strengths. Now, in our case, the coupling strengths are not like in NMR kilohertz, but they are megahertz, many megahertz. And that means if the biomolecule becomes large enough, then you can do this experiment at room temperature. And we have done this with a DNA double strand, which was immobilized on a bead. Now, we saw then immediately that some of the dynamics which we could observe, or let's say the distribution of dynamics we observe at low temperatures, they get averaged now at room temperature. At room temperature, if these dynamics occur faster than our experimental timescale, then we average as everybody does in magnetic resonance. Okay, so now today I will talk about one system, and I will show you what we can learn about the dynamics on the system with this method. And this is it's a simple, small tetracycline aptamer, not too big, so we could actually easily handle it. And there is an X-ray structure existing for this, and this is for us actually an advantage, because then we have a good starting point, and then we can look what we can learn more with the EPR on it. Now, this is an aptamer which is interesting. It binds tetracycline, this small antibiotic here, and if it binds a tetracycline, then the aptamer blocks pre-mRNA splicing, so it can be used for regulation and inhibiting of regulation. And it's artificial one, but it also works actually in cells. Now, this is the crystal structure for us. The starting point here is the binding site of the tetracycline. The structure is solved with the tetracycline bound. There is no structure without tetracycline. And there are actually a number of magnesia ions, double valent magnesia ions, which are important for the structure to stabilize. And so for us, the interesting point was, can we actually confirm the X-ray structure, but then can we also learn something about the dynamics of this complex if the tetracycline is removed, without the tetracycline? And then the other question which I will address, and which we have not published at all, can we also localize the metal binding sites? That is something which is rather difficult in many cases in RNA motifs, but it is important in many cases for the if these are regulatory RNA motifs for enzymatic function. And so to localize that, how many manganese are bound, this is again something which is rather easy in EPR. What you do is you replace the magnesia, which is diamagnetic, by manganese. Yeah? And because the ion radius are very similar, you have to, of course, you have then to look if the function of your motif is still working, which is the case for the tetracycline aptamer. But in many cases, you can do this replacement, and then we have a paramagnetic tag, which we can look at. Now, magnesia is a complicated spin system. It has electron spin 5/2 and nuclear spin 5/2. So the energy diagram is rather complicated, and we have a zero field splitting which smears the spectral lines. So if you have free manganese in water, then it makes a hexa-aqua complex, and then the zero fluid splitting is almost removed, and it gives 6 hyperfine lines which are coming from the nuclear spin. And you can detect it with a CWEPR very routinely at room temperature. Now, as soon as the manganese binds to a biomolecule, then the zero fluid splitting becomes bigger, and then you lose your signal. So what you do is you titrate in the RNA, and then this is you can titrate the manganese or you can titrate the RNA, whatever you want. So here we titrated the manganese. As long as it binds to the RNA, we don't see a signal. And as soon as every binding site is occupied, then you see the increase of the free manganese signal. So without RNA, you see that it immediately starts the signal. If you have the tetracycline aptamer without the tetracycline, then you bind 3 of them, and in the other case you bind 4 of them. You can even see the difference there. So one of the binding sites is related with the tetracycline and needs also the tetracycline. So that is an easy experiment. Now, more interesting now, you have to do pulsed experiments. And one of the ideas is, can we look for the conformational, let's say, for the flexibility of this RNA motif as a function of the divalent metal ion, and also as a function of the tetracycline. And so what we did for that, in collaboration with Snorri Sigurdsson, they put at 2 positions in the stems, you put the spin label. So this is this position and this position. And if you then give the tetracycline and enough of metal ions, then you see exactly this very nice Peldor signal. And here the experiment was done now at a field at Q-band, where you don't get orientation selection, so you can get directly out the distance, and the distance is in very well agreement with what you predict. You predict from X-ray structure 3.8, and we see about 3.9. Yeah, and so a small deviation, which we could also explain, but this is a detail which I'm not going to discuss here. So this is now the similar condition like under X-ray for the crystal. But now what we can do is we can measure now with the ligand and without the ligand for different concentration of magnesia. So we don't take now the paramagnetic manganese, but magnesia. And we just look for how is the distribution in this distance. And if you look with the tetracyclic ligand, you see for high concentration of divalent metals, you get exactly this distance. And then you lower the concentration, and then it becomes suddenly broad. You see a broad distribution, and you see this also as an expert in the time domain. Yeah, here you see the nice oscillation, and then the nice oscillations are smeared out. Now, if you do the same without the tetracycline ligand, then you see a similar trend. So even without the tetracycline, if you go to very, very high concentration, these are non-physiological concentration, but if you go to very high concentration, then the final tetracycline bound state is already preformed. Yeah, it's very, very similar. It's a little bit broader in distribution, but not much. Now, for intermediate concentration, you see that you need actually then the tetracycline to get really the snap-in structure. Yeah, so this is kind of illustrating this behavior for very high metal concentration. You have a preformed state already, but for intermediate concentration of metal ions, then of course the tetracycline results in this structure. You can see this more detailed if you look on the width of this peak, of this distance peak. And so this is the change of the width of the distance peak with magnesium concentration without tetracycline. And then you see the tetracycline makes a big effect, of course, but the effect is almost lost. This is the final structure, the X-ray structure. And you see there is, of course, a difference already here, but the difference becomes much bigger if you have no tetracycline in it. So EPR can be used really to explore the conformational dynamical space of an RNA motif when it binds or not binds to a ligand. Now, I told you one of the metal ions is actually related to this tetracycline binding site, which can be seen here. This is this one. And we were actually interested to see how can we really localize this? Can we really distinguish this binding site from the other binding sites? And what we did for that is, this is a second way how you can work in EPR. You do Endor. Endor means you measure the dipolar interaction now from the electron spin to the close by nuclear spins. Now here, because this is predicted to be very close to the tetracycline, there should be an interaction to the tetracycline. And of course, there should be an interaction to the close by nucleic acids. And so this is the experiment. What we did for this is one of the variants of hyperfine spectroscopy. This is called the Peldor detected NMR, an experiment which was developed in Arthur Schweiger's lab many, many years ago. And it's an experiment where you have two microwave frequencies, no radio frequency, two microwave frequencies. And so you burn a hole in your broad EPR line. And then you detect now the side holes which are created by the hyperfine interaction. And that's so the detection frequency, and that's the pump frequency. And if you look on that, you take the RNA and manganese. Now you replace the magnesia by manganese to see the hyperfine interaction between manganese and the nuclear spins. And then, of course, what you see is you see phosphor couplings. There's no surprise. As soon as a metal binds to RNA, it has an interaction with the phosphor group of the nucleic acids. And if you add the tetracycline, you see some changes, but they are not very dramatic. Now, what we did, and what you can easily do in this case, you take carbon-13 labeled tetracycline, and then what you see is there is a huge peak coming from the tetracycline, which is only arising. So this is natural abundance from the protein, as you see here. But then you see this strong peak, which comes now from carbon-13 labeled tetracycline. Now, with tetracycline, it's very easy because this is, of course, a tetracycline C13 labeled one you can buy easily. So this is nice, but it doesn't tell you the full story. So it does not tell you, you can analyze now these couplings and get distances. But what we saw also, or Thilo Hetzke, who did all these measurements, which I'm showing, what he saw is that, of course, tetracycline is also binding to manganese if there is no RNA around. So there is also an interaction. So it could also be that what we see here is a mixture of manganese binding to the RNA and manganese binding to the tetracycline. So now, of course, with EPR, you can also solve this problem. What you do is, like in NMR, you do a 2D experiment. Yeah? This 2D experiment is now you have two pump frequencies and one detection frequency. And what you do is now you scan now the carbon-13 coupling and the phosphorus coupling, and you look for the cross peaks. And so this is a complicated 2D spectra, which you see here. And but what you see here, these peaks which show up here where I have the arrows, these are the cross peaks between carbon-13 and between phosphorus. So we know actually now that this is really one manganese bound in the same way to tetracycline and on the other side to the RNA motif. So that is something, is a kind of a little bit of a side story here, but I think I wanted also for the NMR audience to say we have these two tools in hand. We can measure the narrow surrounding of our paramagnetic tags, and here if it's the metal centers, this is of course important, and we can measure the long range for which helps for integrated structural biology. Now, then we asked the question, can we also use the Peldor method to measure now these different metal sites here? Again, of course, what you have to do is you have to replace the magnesia by the manganese. And then you put in one nitroxide, yeah, and then you measure all these distances. So the spectra of the manganese is very different from the spectra of the nitroxide. So you can have one pulse on the nitroxide and another pulse on the manganese, and then you can do this experiment. But here, this is, of course, a really challenging task, because you have a number of different binding sites with different distances. So this might become a mess, yeah, because you first of all, again, manganese is a high spin system. It's not so easy. There can be higher harmonics of the dipolar coupling showing up from other transitions, and then you have so many different distances. So this is of course challenging. But what we thought is, or what Thilo was brave enough, and what he tried is, he said, let's do the same like with the titration experiment which we did with CWEPR. And so that is the experiment he did. So he has one spin label, a nitroxide spin label here, and then you do an experiment where you change the concentration of the manganese. The concentration of the RNA was 200 micromolar, and now you have the same amount of manganese as RNA. And then what you see is this upper trace. So these are the time traces on this side, and here you see the distances already. Now what you see here, there is one main distance, there's a shoulder, and then there are some artifacts, because this procedure, how to convert from here to here, is not so easy. And some of the artifacts, they have some errors here. We can even probably explain them. But then, of course, what you did, we searched and we looked what could be the 2.4 nanometer, which we see. And the 2.4 nanometer is exactly the manganese, which is the divalent metal ion, which is bound to the tetracycline. And that is actually known. This is a very, very tight binding. It's a 1 nanomolar binding. So also the affinity of this metal is very, very high. So obviously this is the binding site with the highest affinity. Yeah. And so this distance corresponds very nicely to what we see. Now, if we increase now, we double the metal concentration. And this is the spectra, the lowest spectra. And then we see two distances. And the second distance is 2.2. And then the candidate which was proposed was this is in a loop region where there is a tetra A motif, and it was again proposed to have a strong binding because there are two oxygens which are directly ligated to it. And so this looked reasonable, and so it looks like this binding site is stronger than at the other binding sites because we don't see other distances. If we remove the tetracycline, we see only this other distance. No, because then this one cannot bind, because it can only bind if tetracycline is located. So that looks reasonable, but of course you cannot prove it with this one measurement. Yeah, this is clear, because it's a question of precision, and it's a question of you can speculate. Now, what you can do, of course, you can now go to other labeling positions. So what you do is you triangulate other groups. Olaf Schiemann, Bela Bode, and others have shown to do that. So what you do is now you go to different labeling positions and you do this experiment again. Yeah, and so this is shown a second labeling position. This is a slightly different spin label, but again, it is very nicely confined in its distance and its structure. And if we do the experiment there, we get two main distances. We get some artifacts here, but we get two main distances, and they are in agreement with our assignment. And then we go to another position, and again we get these two distances. So I think what we could show here is that indeed with EPR, with the Peldor method, and also in combination with hyperfine methods, we are able to locate metal binding sites. Yeah, we and other groups have already shown this in other systems, like in proteins, that these measurements from nitroxide to manganese is possible, and you can measure distances. And there are also other methods you can use. You can use the Peldor method or DIA method, but you can also use RITMI for this, which is another dipolar EPR method to use. But for the RNA, I think this is the first example to show that this is possible and that this is working actually very, very nice. Now, it is, of course, also possible, and we have also shown this in model compounds, and others have also shown this, Daniela Goldfarb and others, that you can, in principle, it would also be nice now to measure directly between manganese and manganese. And we didn't succeed to do that on this system yet. We succeeded in proteins to do that. This is a rather demanding experiment because you have broad spectra, you have same shape, and then, of course, if you titrate in with the same amount of manganese, then some of it will be free, and then you will also see a lot of background. And so this is a more demanding experiment. And for this experiment, here these experiments were done with classical pulses, almost classical. The Endor experiment I showed you were with Gaussian pulses, but beside of that, everything rather classical. In the other case, you have to use broadband shaped pulses. And there's another topic. I'm not talking about that today. Okay, so I hope I showed you on this one example that pulse DPR spectroscopy in the combination with Endor is able to measure structure, but more importantly, I would not so much claim on the structure, but the conformational flexibility of RNA motifs as a function of their interaction with other proteins, DNA molecules, or ligands, or something. And I think this is interesting, and we can also use this method to localize metal binding sites, and we can do this, of course, also in proteins. And I think that is an important biological tool for biological structure investigation or functional investigations. And so I hope that is the take-home message. That is something like you have transporter proteins which have a nucleotide binding domain with magnesia and something like that. Then of course you can use these methods to investigate in detail what they are doing within function. The experiments which I have shown here, they were all done by Thilo Hetzke, a very an excellent student, PhD student in my group, and he's now with Bruker Biospin. The collaboration were with Snorri Sigurdsson for the spin labeling, and with Beatrice Julia Weigand, Mark Vogel for the construction of the aptamer, and Alice Bowen was a postdoc in my group involved in the 2D hyperfine Endor experiments at that time. She's now University of Manchester. And here are the funding. We have a collaborative research center on regulatory RNA here in Frankfurt, headed by Harald Schwalbe, which is very nice for all these collaborations. And of course, thanks to our Biological Magnetic Resonance Center here in Frankfurt, which is always supportive for instrumental repair. Yeah, and of course the university. And of course I thank you for your attention, and I'm happy to answer your questions.
Okay, thank you very much, Thomas, for the great talk. And I see already some questions. I will start reading from the Q&A. So
Marina, can I interrupt as a
Sure.
panelist while the questions fill up in the Q&A and ask two
Sure.
quick questions? Harold must have NMR spectra of the aptamer as a function of magnesium with and without tetracycline. And frequently these aptamers without their ligand give very disordered looking NMR spectra. Your distance measurement might suggest that at high magnesium, you would see a structured NMR spectrum. Is that the case?
Yeah. So for this system, we didn't join up with the NMR groups yet, I have to say, but we have measured a number of different aptamers, and one of the aptamers, to give you an interesting, this is a very interesting aspect. One of the aptamers was a neomycin binding aptamer, and Jens Wöhnert has done the structure, the NMR structure on it. Not Harald, but Jens has done the NMR structure on it with the neomycin bound. And we did the EPR with the neomycin and without neomycin, and we found the same structure without and with neomycin. And we were really worried about that, because Jens, of course, says if the neomycin is gone, the structure loses all the contacts. Yeah. Now, in this case, in the neomycin aptamer, this is an example which is a little bit exceptional of what I said before, that we freeze the conformational space. What happened there? And then we figured out together with Jens, if you cool down your system, then you reduce the entropy, and then the neomycin aptamer is very flexible at room temperature. And then if you freeze it, it freezes actually in the preformed ligand-bound state. And Jens could actually follow this exactly, measuring now the NMR spectra at lowering temperatures. And so in this case, what we see in EPR is just the energetically favored state. Whereas if you go to room temperature, the entropy wins, and then you see the broad state without the ligand. With the ligand, of course, it's a very structured state. So that is always something which we have to keep in mind, that if we freeze down a sample, we might not see what is happening at room temperature. For this, we can now use also this Peldor method with this rigid spin label at room temperature, because you can measure at room temperature. You just immobilize the rotation of the RNA molecule, but we don't have to freeze it. Yeah, and you do this at room temperature, and then you can make sure that what you see at room temperature is the same what you see in the frozen state.
And then if I could ask one technical question, you said you didn't want to talk much about the shape of the pulses and their bandwidth, but do you have to take the excitation profile of the pulses into account when you calculate the Gere curves?
Yeah. So these quantitative simulation I showed you, you only achieve them if you understand the shape of your pulses in every detail. Yeah, and for that reason, of course, nowadays shape pulses are much advantage, because we know exactly which spins we excite. Yeah, and if you do this, you can calibrate this. You can internally calibrate this, and then you can, if you have a structure, you make a prediction of the time trace. And you don't have to fit the distance distribution, you fit the time trace directly. And if you have a structural bundle, you can make a direct prediction of how the time trace looks like. And this is just an addition of these different structural ways. And there is almost no free parameter in there. Yeah. But you have to understand your pulses, and you have to set them in the right way, and you have to know that. But the EPR community now knows that very well. So we are in a good position there.
Well, if this was in person, I would take you for a coffee and ask you an hour's worth of questions, but I better let Marina take over.
We can have a chat, a private chat after. Yeah.
Okay. All right. I see here a question by Thomas Schmidt, and he says, or he writes, "Great talk. I appreciate the idea of spin dilution with manganese. Did you look in using different pump pulse strengths instead? The benefit would be also that one could observe the number of spins coupled to the nitroxide based on the relative modulation depth."
Sure, we are working on that. I have seen before the talk was starting, I was not expecting that, but I have seen that there are really a number of EPR people in the crowd. And then I regretted not to have talked about shape pulses and pulse sequences, but I thought I will lose a lot of the NMR people, the biological NMR people, if I do that. So of course you can do that. And again, quantifying with manganese is not so trivial. So we are trying to do this now with model compounds, and it's on the way. And that would be great. I think it could be an alternative to do it with the CWO, and maybe more precise.
Okay, then there is a comment by Angeliki Giannulli. She writes a beautiful MNO manganese nitroxide data. And then QA is still open. Maybe I would ask a question in a more general direction. Thomas, you mentioned you can do this experiment, or you've done experiment also in cells, also with RNAs. And what spin label did you use? What is your experience going in that direction?
So in the cells, as the EPR people know, the classical nitroxides with the methyl groups, they get reduced in a rather short time. In the best case, 20 minutes, half an hour. And that means you have to freeze the sample very fast. And that is a little bit of a problem, because if you bring in the spin labeled molecules in the cells, then you have to heal the cells, and it takes some time. And then and this is not optimum. But what you can do is you can replace these methyl groups now by shielded groups like ethyl groups, and then the spin labels survive for a very long time. So again, fortunately, but this is again the topic which Alberto will talk next week in detail about. We have this shielded nitroxide, so we don't have to worry. And then we have measured with flexible spin labels. We have also measured with rigid spin labels in the cells, and that of course helps you to get really details about structural changes, which you see. Yeah. We have published this, as I showed in the beginning already in Angewandte, that we see even on a double-stranded RNA, we see changes. Yeah. And the molecular dynamic people, they can at least support a little bit that, because RNA is highly charged, and then you get interaction with proteins, of course, and then it starts to wiggle on the RNA, and then you see, of course, some changes. They are not really dramatic, but our precision is high enough that we can distinguish.
Yeah. So basically, when you do the Peldor, the distance measurement, you freeze the sample anyhow. So when you are in the cell, it's dead enough that you say it's the spin label cannot reduce at room temperature, and one that is frozen is stable.
Yeah.
So okay, then.
So as you know, but usually, let's say you want to have the molecules distributed in the cells, and then you want to be that the cells are everything is fine. So it's better to have these protected spin labels. But again, they exist nowadays. You can also use, like Elena has shown and has demonstrated, Olaf Schiemann, you can use tritial spin labels, which are very stable in cells. And Daniela has shown, she will also talk next week on this in-cell workshop, with gadolinium. Of course, you can also use gadolinium. We have not used gadolinium with that yet, and we haven't, but the others have. Olaf has done, and Elena has done in-cell measurement with tritial spin labels, and Daniela has done a lot of in-cell measurements with gadolinium spin labels. So we have a number of different labels with different advantages and disadvantages, but which are stable in cells. Yeah. And the sensitivity in EPR is high enough that we can do that. Yeah. We can measure a 50 micromolar concentration, and if you go to higher and higher fields, our samples become smaller and smaller. Our sample volume, if you go to 260 gigahertz, as the EPR people know, but the not in EPR people don't know, is something like 20 nanoliters. Yeah, and that means a few cells. Yeah.
Yeah.
Thomas, I have a question. Like
sure.
in this manganese titration, I find that very intriguing. So you associate to the highest affinity because you saw only one band. But then my question is, how much different do you know it is the affinity of the first site with respect to the second site?
Yeah. So we didn't analyze that in detail yet. No, in principle, what you have to do is now number of concentrations, and then you have to you have to get these peak heights. You can also compare with the CW and analyze this curve in detail. We didn't do that yet. So I'm not sure if you will get really very hard numbers for that, but what you can say, you can get an ordering. You can say this is the tightest binding site, the second one, and I don't know how further you can go.
Mm-hmm. Oh, that is very cool. Like because you can solve them out, and
I may just quickly jump in. I mean, let's maybe stop the recording and proceed to the unofficial part, and then we can ask all the other questions and handle them. So.